One of the foundation stones of modern Darwinism is the Weissmann barrier, named after this man, August Weismann, described by Ernst Mayr as the greatest evolutionary thinker of the 19th century, save Darwin himself. The Weissmann barrier supposedly demolished forever the Lamarckian idea that adaptation in individuals could affect adaptation in evolving lineages. Once the Weissmann barrier went up, no longer could there be any talk of giraffes stretching their necks or of strong-armed babies of blacksmiths. There could be only one true theory of evolution: natural selection of apt-function genes. There's a problem with this story, though. It's largely untrue. By this, I don't mean to say that the Weissmann barrier does not exist, only that it has taken on a meaning that is quite different from August Weismann himself thought it meant, and, as it turns out, what modern findings in genetics are telling us about how it works. The Weissmann barrier is also known as the segregation of the germline. This means that in the living body there are two populations of cells. One, the soma, constitutes the body itself: the skin, bones, muscles, organs, and so forth. Another, the germline, comprises cells that are set aside early in embryonic development and are destined to become the gametes, the eggs and sperm. Germline segregation asserts that the soma can sustain the germ cells, provide them nutrition, a place to develop, and prepare them to combine with other gametes. But the soma cannot affect the nature of the heritable stuff in the germline cells. This negates the Lamarckian idea that adaptation in the soma, physiological adaptation, can somehow be imprinted onto the gametes. The variation that natural selection needs to operate can therefore only originate in the germline. Weissmann's barrier is made all the more compelling because it supposedly is bolstered by a decisive experiment. Like all good experiments, Weissmann started with a question: what if Lamarckism was true? If so, then there should be some effect of repeated alterations of the soma over many generations. To test this idea, Weissmann took a colony of mice and amputated their tails. He then bred these mice with one another and docked the tails of their offspring. He then bred these mice and docked their offspring's tails, and so on for more than 20 generations. If Lamarckism was true, the tails of the mice in the 20th generation should have been shorter than the tails of the mice in the first. This was not the case. The tail stayed as long as ever. QED, Lamarckism is disproved. This experiment has been cited as one of the greatest biology experiments of all time. Well, it was a very good experiment, but it wasn't that great. Is mutilation a good model for natural adaptive variation in the soma? Hardly. Was 20 generations enough to demonstrate an effect? Who knows? What's most interesting, though, is that the Weissmann experiment has taken on a kind of legendary significance that Weissmann himself never really attributed to it. In fact, it does a bit of a disservice to August Weismann's real importance to evolutionary thought. So let's try to peel back that myth and get to the real story. To understand Weissmann and germline segregation properly, we need to be mindful of how people then thought about heredity and its relationship to evolutionary change. First, it was commonly believed that the hereditary stuff was some sort of hereditary fluid. This is the origin of words like germplasm, its literal meaning is germinal fluid, and cytoplasm, and common phrases like bloodline or in the blood. This belief arose from a deep reservoir of folk wisdom, natural history, and husbandry, because a fluid heredity is a very natural metaphor for a common observation: traits blend. How else, but with a hereditary fluid that could blend, could one explain why offspring bore some mixture of their parents' heritable traits? Or how domestic livestock or crop plants could be improved by blending desirable traits from different stocks? Second, heritability was thought to exist in two forms: soft inheritance and hard inheritance. Hard inheritance was thought to be what kept lineages from jumping the tracks, so to speak, why dogs always have puppies and why cats always have kittens, but dogs never have kittens, or vice versa. The problem with hard inheritance, though, is that it is conservative, and this means that evolution could never happen if hard inheritance was all there was. This is where soft inheritance comes in, which introduces the variability that is needed for selection to work. Why one can draw, say, various breeds of dogs out of the basic dog lineage. Soft inheritance was also thought to be involved somehow in physiological adaptation of the organism to environment, which could be transmitted from generation to generation. This belief was the heart of the implicit Lamarckism that pervaded Darwin's own thinking about evolution, and Weissmann's too. In Weissmann's time, in the late 19th century, this scheme of heredity was proving to be problematic for the Darwinian idea, and this precipitated the so-called crisis of Darwinism. Blending inheritance, which follows naturally from the idea of a hereditary fluid, was the main problem. Let's illustrate. Imagine that a striped zebra shows up in a population of plain-colored animals. When the striped zebra bred, it would most likely be with a plain-colored mate. Blending of their traits would produce offspring with less distinct stripes. When these mated, the stripes would fade further still, and so on until the new trait of stripiness was swamped in the sea of blending inheritance. You can see the problem. Natural selection could not work if every new trait that arose was swamped away by blending inheritance. Here's where August Weismann came to the rescue. Microscopy at the time was progressing in leaps and bounds, and this made it possible to observe the goings-on in cells with a detail that had never been possible before. Weissmann was one of a group of German biologists that then were at the forefront of this new science of cytology. And what they were finding was proving incompatible to the idea of fluid inheritance. For one thing, there was a tendency of the stuff in the nucleus, whatever it was, to coalesce into discrete bodies whenever the cell divided. We now call these the chromosomes, of course. And then there was the odd observation that these chromosomes, whatever they were, would be partitioned precisely in half whenever gametes were formed. This we now know as meiosis. It was not Weissmann himself who carried out these observations. He suffered from an eye disease that kept him away from the microscope. But it was he who melded these observations into a new theory of heredity. What if the hereditary stuff was not a fluid, but consisted of hereditary particles? That way, new traits, which amounted to changes in these hereditary particles, could not be diluted away, but could be carried unchanged from generation to generation. If this sounds like our modern conception of the gene, you'd be correct. Weissmann's so-called particulate theory of inheritance was the starting point for modern gene theory, and this was his most important legacy to biology, not some gimcrack experiment chopping tails off mice. It was significant also because it got Darwinism over the potentially fatal crisis of blending inheritance. There was another crisis lurking for Darwinism, though, this one over the relationship between soft and hard inheritance. That crisis came to a head mainly at the instigation of this man, Thomas Hunt Morgan, who was well known to every biology student that ever bred a fruit fly. Morgan, of course, is the biologist who adapted Weissmann's particulate theory of inheritance to the reality of the chromosome, a string of particulate genes in linear array along chromosomes. What's interesting about Morgan is that he was a staunch anti-Darwinist and a harsh critic of Weissmann's too. There's a historical irony in there that we must understand because our modern conception of Darwinism as selection of genes for apt function traces ultimately to Morgan's fundamentally anti-Darwinist theories of evolution. Morgan championed a model for evolution that he called mutationism. By this, he meant that the variation that drove evolution came in the form of sudden and random mutation of genes. Adaptation, that fundamental kernel of the Darwinian idea, had little to do with it, or was, at best, only a minor factor. Morgan had a lot of good science on his side of the argument. Mendel's experiments on peas had finally emerged from their 35-year slumber, confirming that inheritance was indeed particulate and that genes were those hereditary particles. Morgan and his students could point to obvious, visible mutations in his fruit flies that arose spontaneously and were heritable, consistent with Mendel's conception of the gene. Finally, Morgan brilliantly reconciled the Mendelian gene with seemingly anomalous behaviors of the chromosome. For all this, Morgan was awarded the Nobel Prize in 1933. There's no doubt he deserved it. But Morgan also went a little too far. Morgan's beef with both Darwinism and Lamarckism was their notion of adaptation, which, as I've said, was built primarily around the idea of soft inheritance. Morgan's work had convinced him that there was only one kind of inheritance, the hard kind, and that was the mutatable gene. Adaptation, therefore, could only arise from a lucky mutation that improved function and survivability. Because Weissman was a Darwinist, and therefore a devotee of soft inheritance, Morgan regarded most of Weissman's evolutionary thinking to be suspect. But there was one aspect of Weissman's thought that Morgan found attractive, and that was germline segregation, which Morgan thought was essential for his mutationist theory to work. And so he elevated Weissman's idea to the level of a general principle of evolution, an impenetrable barrier between physiological adaptation and evolutionary change. And what of Weissman's experiments with mutilated mice? That was useful to Morgan too, but for a different reason, because it handily demolished the other main competitor to his mutationist theory of evolution, namely Lamarckism. This is how this experiment, so trivial to Weissman himself, came to assume its legendary status today. The foundation laid by Morgan is usually cast as the final resolution of the 19th century crisis of Darwinism. Because Morgan was not a Darwinist, it was obviously nothing of the sort. It was the triumph of the fundamentally anti-Darwinist school of mutationism. To call it neo-Darwinism, as we do today, is a bit of a distortion. If anything, the crisis of Darwinism persists to this day. However, new discoveries in genetics are arising that just might finally resolve that crisis. And the resolution might come by casting the Weissman barrier and germline segregation into a very different light from what we're accustomed to, something closer to Weissman's own conception of it. Within a developing embryo, there's an evolution of sort that takes place. A genetic founder, the zygote, divides repeatedly to populate the body with the several trillion cells that come to compose it. As these cells divide, they differentiate, evolving to become the various types of cells in the body, about 210 distinctive types by the latest official count. All these cells contain the same suite of genes the zygote had, but as they differentiate, each cell type comes to be marked by the silencing of different suites of those genes. Brain cells, for example, silence a different suite of genes than do, say, muscle cells. There are many ways these genes come to be silenced, but one important way is to attach a methyl group to a nucleotide base. This is called DNA methylation. When a patch of DNA is methylated, the proteins responsible for gene transcription cannot get to it, and so the gene cannot be expressed. As the embryonic cells differentiate, each cell type methylates DNA in a different pattern. One pattern might mark brain cells, a different suite marks muscle cells, and so forth. The most interesting thing about this methylation process is that it is heritable. That is, a patch of methylated DNA, when it replicates, will produce a methylated daughter DNA that has the same methylation pattern as the parent DNA. In the context of embryonic development, this means that differentiation is heritable. More broadly, though, heritable methylation patterns mean that there are at least two forms of hereditary memory. First, there's the well-known memory embodied in sequence information in DNA molecules, the standard conception of the gene as a DNA nucleotide code. This is called genetic inheritance. The second is the heritable pattern of DNA methylation. This is called epigenetic inheritance. And it is, provocatively, the very thing that modern Darwinism holds to be impossible, the inheritance of an acquired character. But let's remember that inheritance of acquired characters is a caricature of the Lamarckian idea. What Lamarckism really asserts is a fundamental similarity between adaptation in the individual and adaptation in the lineage. In other words, what governs the evolution of cells in the embryo can govern the evolution of lineages. This is where things begin to get very interesting, because DNA methylation doesn't operate only in embryonic differentiation. It is involved in physiological adaptation as well. We know, for example, that different diets can produce different methylation patterns in the numerous genes that control metabolism. These, in turn, can have long-lasting effects on how the body handles food. Should it be stored as fat or burned off as it is taken in, for example? The Lamarckian idea, the real one, not the caricature, could be credible if these secondarily acquired patterns of methylation could be transmitted through the gametes. This brings us back to Weissmann's segregated germline. Lamarckism would be falsified if, for example, the methylation patterns of the cells that are destined to become gametes were erased, hitting a genetic reset button, so to speak. There, not in tails of mutilated mice, is our falsifiable test of the Lamarckian idea. So what happens to the methylation of germ cell DNA? In all animals, both sperm and eggs develop an intimate association with the somatic cells that compose the gonads. These somatic cells control everything about the progress of diploid germ cells to haploid gametes, including patterns of DNA methylation. In sperm, for example, the DNA comes to be heavily methylated, in fact becomes the most heavily methylated genome in the body. This is probably secondary to the need to silence the sperm cell's genes so their DNA can be packed tightly into that little genetic torpedo that is the sperm. Once the sperm nucleus penetrates the egg, its DNA is largely demethylated, so there's a genetic reset of sorts that takes place. But here's the interesting part. It never becomes completely demethylated. There is always some residue of methylation imprinted onto the sperm DNA by the somatic cells in the testis. Something similar happens in the ovum. During the final stages of ovum maturation, the ovum genome becomes largely demethylated, but again, not completely so. What's most interesting is that the egg's DNA becomes partially remethylated prior to ovulation. All of this is under the control of somatic cells that surround the germ cell and guide it on its way to becoming the gamete. This includes controlling what patterns of methylation are imprinted onto the haploid DNA. This points to a quite radical conclusion. Sequestering of the germline does not appear to insulate the germline genome from the experience of the somatic genes, remembered in acquired patterns of methylation. Rather, segregating the germline appears to do precisely the opposite. It actively manages how the somatic genome imprints its adaptive experience onto the germline cells. This puts Weissmann's evolutionary thinking into an entirely new perspective. Despite its billing as a decisive disproof, germline segregation has always been a pretty weak critique of Lamarckism. As I've said elsewhere, it applies only to a limited set of organisms, the animals. Plants have no identifiable germline, and nor do any of the other eukaryotic kingdoms, like the fungi or the protists. And it doesn't apply at all to the multitudinous and diverse bacteria. Even among the animals, it only applies to sexually reproducing animals. And even among them, there's a great deal of diversity in how and when the germ cells become a distinctive clone of cells. August Weismann was no fool, though. Weissmann came to the idea of the segregated germline through careful study of a wide variety of organisms, and saw germline segregation for what it was, an evolved trait of a specific and limited set of animals. The simplistic view that germline segregation was a general principle of Darwinism was, rather, built up largely by Thomas Hunt Morgan, who, as I've said, was no friend of Weissmann's. Morgan, in fact, trivialized Weissmann's thought to support his own anti-Darwinist model for evolution. For most of the 20th century, it was Morgan's, not Weissmann's, conception of the germline segregation that prevailed. The rising science of epigenetics is unsettling all this, and in doing so is making new sense of the subtle Lamarckism that pervaded the thinking of the classical Darwinian idea, that adaptation, not mutation alone, drives evolution.